Thank you very much. All right, good afternoon, everyone. So very excited to introduce our speaker today, one of our newly minted professors in the department who just joined us back in July of this year, Yaxi Hu. So Yaxi got her MSc and PhDs from UBC, British Columbia, BSc in Biological Science from China Agricultural University, and then did postdoc trainings in the Chem department of UBC, as well as the Food Research Division of Health Canada. Her research program is focused on developing analytical tools for food authenticity, quality, safety issues, also looking at analytical methods to detect food safety quality as well. The analytical methods developed in Yaxi's lab range from cheap and user-friendly sensors to highly accurate, sensitive, more methods for confirming analysis using sophisticated instrumentation. So today we're going to be hearing about, let's move my window here. So advancement of food techniques for food authentication, the last barrier to prevent food fraud. And thank you so much, Yaxi. We look forward to hearing this.
Thanks, Jeff, for the kind introduction, and thanks, Maura, for organizing this Science Café and inviting me to give this talk. Good afternoon, everyone. It's my great pleasure to be here presenting part of my research project regarding the advancement of techniques for food authentication, the last barrier to prevent food fraud. I'm Yaxi Hu. I'm an assistant professor in the chemistry department at Carleton University. So just a brief introduction about myself. So I was born and raised in a city where it hosted both the Summer and Winter Olympic Games, right? It's Beijing. It's a great city with a lot of historical sites of China, but also it's a great showplace of the modern China. And I hope when this pandemic thing is chilled down a little bit more, you will all have a chance to visit this beautiful city. And I believe, I'm pretty sure it won't let you down. About my training background, Jeff already introduced that part. So basically, I had a mixture of background in the biological science and then in food science before I joined Carleton University in the chemistry department, and I'm specifically in the food science program here. So I assume some of the audience here today don't really know what is food science. You don't have a food science background. And I know that some of the people think we are kind of like the chef. Well, I'll say there are some overlapping perspectives regarding food scientists and the chef, or in general, the catering services, but there's also a lot of difference. So for the catering and the chef, they interact more directly with the endpoint consumers in the restaurant. But as food scientists, we interact more often with the food, with the products, with the material. So everything related to transforming agricultural product into a final food products is covered or can be covered by the food scientists. And it's a really interdisciplinary area, which include the chemistry, the biology, microbiology, processing, packaging, safety, quality, and integrity. And among all these different areas, the one that I'm most interested in is regarding the food integrity. So what is food integrity specifically? When we want to secure the integrity or ensure the integrity of the food products, we want to make sure the food is free of these four major issues. They are the food quality issue, food safety issue, food fraud issue, and food defense issue. These four issues have some inherent differences. So for the food quality issue and food defense, food safety issues, these are generally naturally occurring defects that's not caused by any human intentional practice. On the contrary, for the food fraud and food defense, these are usually intentional practice. So there are people intentionally modify the food for some certain purpose. So for the food quality and food fraud issues, they're usually not causing any illness. And food fraud as an intentional practice, the motivation is to get extra economic benefit. While for the food safety and food defense, these issues usually can cause the illness of consumers. And food defense specifically, the practice is to damage the consumer's health. In my research lab, I deal with the issues regarding the safety, quality, and fraud. But for the sake of time today, I will only be talking about some of my research I did in the food fraud area. So if you search Google, do a simple Google search for food fraud, you may find a lot of alarming news. So this one, this is an operation done by Europol and Interpol, the police officers. It was back in 2021. They did a 6-month operation, and across 72 countries, they found more than 15,000 tons of illegal or fraudulent products. That's a big number. Then during this several years of the pandemic, because there's a lot of fluctuation and disturbance in the global food supply chain, food fraud is believed to be more severe and especially harming the smaller business. Some more specific food fraud cases. So the European study found adulteration in many herbs and spices products. So a lot of different extraneous materials being added into these products to increase the volume. This melamine case happened in China in 2008 was another big one. So this one caused thousands of injuries and death of baby in China. This case was thought to be a food safety issue at the beginning because there's so many sickness. But after people looking at the motivation behind it, people found it's actually a fraud. So melamine was added to increase the apparent protein concentration of the dairy product to make extra economic benefits. Horse meat scandal is another big one. This one initiated in Europe. So horse meat was added into ground beef burgers without labeling on the package. But because of the international trading, North America was also, you can find like consumers in North America was also hurt by this scandal. Lobster balls doesn't contain lobster, and beef balls contains pork and chicken. It's another very typical food fraud. Fake honey. So honey, as one of the most expensive sweetening agents, has a high chance to be adulterated using some other cheaper syrup or just add some sugar in it. Pomegranate juice is another possibility. Pomegranate is a superfruit with a lot of health benefits. But pomegranate, the production cost is relatively high, while the production yield is relatively low. So there are chances for fraudsters to manipulate the product and to reduce the cost. This is a map showing, so summarized by Oceana in 2016, so showing the global seafood fraud and mislabeling status. So all of the fish dots you see on this map represent a study that found more than 50% of the fish being sampled in that area were mislabeled or fraud. So in general, it's estimated that food fraud is costing the global food industry $65 billion per year. And about 10% of the food products are impacted by food fraud. So by summarizing previous studies and reports on food fraud issues, they've identified some foods that are more vulnerable to food fraud compared to the others. And here are the top 10. So they're the olive oil, fish, organic foods, milk, grains, honey and maple syrup, coffee and tea, spices, wine, and certain fruit juices. And here are the several major types of food fraud practices. They are the dilution, which is a process of mixing an ingredient of a higher value with an ingredient of a lower value. And substitution is a process of replacing a nutrient or an ingredient or a food with another one of a lower value. Consumer, hiding a low quality food or ingredient with or selling it as a more premium product. Unapproved enhancement is a process of adding unknown or undeclared compounds to food products in order to enhance the quality attributes. Counterfeit is more about intellectual properties. Mislabeling, this can happen intentionally or unintentionally, depending on if the manufacturer knows intentionally did it, or like knows they did it wrong or right. Sometimes mislabeling can happen when the manufacturers lacks that part of knowledge. But regardless of what, so mislabeling is related to the false claims or intentionally distortion of the information provided on the package or labeling. Free market forgery is another type of common food fraud practice. So if we look closer at the most vulnerable food products and the common types of food fraud practices, we can find some trends or some common features regarding why certain foods are more vulnerable. And here are the 4 features that we summarized. So usually the food needs to have a high margin benefits for the fraudsters to want to manipulate that part, food product. In the case of wine and maple syrup. Some other products, they have a high production cost, low yield, but also in a high consumer demand, such as the pomegranate juice. Extensively processed food, of which the original physical and morphological features being completely removed, are also more vulnerable to food. For example, the spice powder, fruit juice, and fish fillet. So imagine if you're purchasing the spice, the whole seeds or the whole fruit, and also the fish with the head, the tail, the bone, the skins, everything on it, it's less easy to replace it with something else. Another type of food that are also vulnerable to food fraud is some of the food, their selling point are focusing on the features that are not visible. For example, if you're purchasing the products that with a specific geographic origin, or you want a product that's farmed using a specific method, for example, the organic versus conventional. These features are not visible directly to the consumers, so there are chances or they can be easily mislabeled. It seems like based on the definition, food fraud is only harming the economic benefits of consumers, but actually that's not true. There's more damage food fraud can do, including the economic loss of all the stakeholders, such as the genuine food industry and the consumer, as well as the government. Once the food fraud case being reviewed, consumers' trust in the food industry and government will be weakened substantially. And it could take years to regain that part of the trust. What's worse with food fraud is there are potential health risks related to it. So the melamine case is a great example for this. So the poisoning chemical melamine was used as the adulterant without knowing the potential harmfulness on human. And there's also other cases regarding the incorporation of allergens. So there was a case several years ago. So spice powder was mixed with peanut skins, and peanut is a very common allergen in North America. If consumer consume this spice powder without knowing there's a peanut present, they will be in trouble. Pathogenic bacteria can also be incorporated into the product if contaminated ingredients are sold as a premium ones. So I'm not sure if you're aware of this. We don't hear the word food fraud very often, even just a couple of years ago. But food fraud is actually not a new issue. So this is a very rough timeline about the recorded cases of food fraud happened in the history. So dated back to the 8th century BC, there were records showing that ancient Rome wine, the wine was adulterated using lead as a sweetener. And lead is a heavy metal that everyone wants to avoid. And then in the 10th century in Europe, there were laws available to punish the selling of adulterated food, which means selling adulterated food is not a minor issue at that time. And then in the 14th century, 16th century, bread was found to be adulterated using some exterior materials to increase the volume or weight. And in the 18th century, there was also laws to punish the selling of fake tea Another case in the 19th century, peppercorn and powder were found to be adulterated with linseed oil and dust. And then coming closer to modern, in the mid 19th century, New York residents were purchasing the swill milk made with even unedible chalk and plasters. I believe these cases are just the tip of an iceberg, but I also believe that with the current situation of the global, highly complex global supply chain, food fraud is getting worse. So to have a better understanding or better idea of how complex the supply chain can be, here is an example of a global seafood supply chain. So in this case, the catcher, when they catch the seafood, they can sell it directly to a processor inland, or they can sell it to another vessel or at-sea processing before this pre-processed food, seafood being shipped to the processors on land. And then these products being processed can be purchased by the distributors in another continent or in another country, and going through several different levels of distributors before they eventually go to the hand of consumers through the food service or restaurants or grocery stores. These different steps can involve several different countries. One of the examples I heard is, so for example, the fish is harvested in the Pacific Ocean near Canada, and then it can be sold to China for filleting. So to remove the bones, remove the skins, and the fillets of the fish can be sold to the US for breading and frozen. And then that product could be shipped back to Canada again and sold in the Canadian market. But then according to the current labeling requirement, this product only needs to be labeled as a product of the US. So the information about it's being harvested in Canada and being processed in China, all this information will get lost. So these gaps in the regulation and also the complex global supply chain have really created a lot of opportunities for the fraudsters to manipulate the products. So to address food fraud issues, one of the best strategies is to establish effective traceability system. This is a great preventative strategies. And with all the detailed information being recorded along the supply chain, there will be less opportunities for fraudsters to participate. So there have been a lot of discussion and reports regarding how the food traceability system can help prevent food fraud. But if we look at the high prevalence of food fraud, we can easily draw the conclusion that traditional traceability systems are not providing a very effective protection to the food authenticity. So to find a better solution for food fraud prevention, we need to understand what are the challenges faced by the industry. Here are several challenges that we identified. So definitely the complex global supply chain really complicates the information sharing system and impair the visibility of all the stakeholders into the supply chain. And also, it's a common issue in the food industry, especially for the smaller size industry. The traceability information are still being recorded on paper instead of digitized, and making the tracing of that information very time-consuming and labor-intensive. What's more difficult for effective food fraud prevention is that even though some of the industry, they are doing a good job for sharing the information along the supply chain, it's very difficult to verify the information. And for most of the time, people are doing this by trust. So they're trying to identify reliable suppliers. But this is also a vulnerable part that the fraudsters can take advantage of. So to better address this issue, we need the help of reliable authentication tools to provide the last barrier of protection in the case, especially in the case that the traceability system are not functioning properly. So traditionally, authentication tools are usually used as an endpoint analysis to prevent fraudulent products to go into the market. But on the other hand, some of the authentication tools can be applied to monitor the authentication information of a product in the real time along the supply chain. And then if combining with a digital traceability infrastructure, which allow the information to be shared in a timely manner, we can make the traceability system very smart. And hopefully this smarter traceability system can better safeguard the authenticity of our food products. So I was trained in the food science and analytical chemistry. Part of my research at Carleton University is to focus on the development of effective and efficient analytical tools, and to combine them with the Internet of Things to ensure food authenticity along the supply chain from the farm to the fork. And to ensure a smooth integration of analytical tools and the IoT, we also need the help of advanced informatic tools, such as the machine learning, cloud computing, and artificial intelligence. So now I'd like to talk more details about how we are developing these new methods, new analytical methods to tackle the food fraud issues. And from this slide on, my talk will be a little bit more technical. So to design or select proper analytical method, we need to know what are the targets we're trying to analyze. And in the case of food fraud, the type of undeclared materials can be these three categories. So they can come from a different biological species, or from the same biological species, but a different part of that species, or also it can be from a specific chemical. Then, depending on the type of adulterants that we're analyzing, we have to select the techniques accordingly. So currently, the commonly used techniques can be grouped into two categories. One is based on the DNA sequence or genetic code, and the other is based on the chemical composition. So depending on the adulterants, if you suspect the adulterant, or in the specific case of food products, the common adulterant is coming from a different biological species, for example, the horse meat and the beef meat, there are differences in the genetic code. So we can use a DNA-based method for the analysis. And because they are different in the genetic code, their downstream small molecules or overall chemical composition will also be different. So there are chances for us to use a chemical composition-based method to differentiate the adulterant from the genuine product. But if the adulterant is coming from the same biological species, for example, a different meat cut, or from a specific chemical such as melamine, then the only solution will be a chemical composition-based method. So currently, the gold standard methods for the DNA-based analysis is the PCR. I think after this several years of pandemic, a lot of you have at least heard many times about this PCR technology. So this is a technique to amplify the DNA or RNA in the biological species, and then combine it with the result visualization method. You will be able to see which biological species is present in your sample. Well, for the chemical composition-based method, the most commonly used one is chromatography. Liquid or gas, either one are commonly used. So chromatography is a technique that is basically trying to separate each individual compound for you to analyze them. Instead of, because for the food sample, you can have thousands of different chemicals in that one sample. And it's almost impossible if you don't do any separation and you want to analyze everything all together. These methods are they're very well established and can provide reliable results, but there are some disadvantages for them to be used for the smart traceability system that I'm interested in developing. The disadvantage, including they're relatively time consuming, requires quite extensive sample preparation, quite complex procedure to complete the whole analysis. So we need highly trained personnel. And the capital cost and also cost per analysis is quite expensive. And what's more is that the instruments are generally bulky. It's not very feasible if we want to do this analysis for the in-field or online analysis. So to fit our purpose of developing the smart traceability system, we need authentication tools to be simple, rapid, cost-effective, and portable. So in the following slides, I'll be introducing two authentication tools that I developed that can be integrated with the advanced traceability system to allow a smart traceability. So the first study is authentication of the fish species. In this study, we used a portable Raman spectrometer. So the instrument is of a size similar to a computer mouse. And for the Raman spectrometer, you can imagine this technique as a specific type of camera. So camera is recording the information of the visible light, while the Raman spectrometer is recording the information of another, of a different wavelength in the electromagnetic radiation spectrum. So in this study specifically, we developed an analysis to differentiate 11 species of fish. So the first step for this study was we collected the Raman spectra for this 11 species of fish and constructed different mathematical model for the classification. And then eventually developed this hierarchical structure for the analysis. So the next time, if you have a new fish sample, you want to identify the species, you only need to collect the Raman spectra of it. And then the first step will be to identify if it's a Salmonidae fish or non-Salmonidae fish. This step can be done by a visual analysis, but if you want to make everything more pipeline or more automatic, there's also a mathematical model that can do this differentiation with 100% accuracy. And if the fish sample is identified as the Salmonidae, then we have another math model to tell you which specific type of salmon it belongs to, with 88% accuracy. But if the sample is a non-Salmonidae sample, we have another mathematical model to tell you with 91% accuracy which type of fish it belongs to. So currently, the gold standard method for fish identification is called DNA barcoding. It's using a PCR method and combining with a sequencing method to tell you, and then match the result with the database to do the identification. And usually that analysis can take up to 2 days to get the results. And involves a lot of complicated experiments, including the sample, the DNA extraction and preparing of a very complex reaction system. Well, for our Raman spectrometer-based method, we only need 5 minutes to tell you the result from the spectral collection. So you can see that for this analytical pipeline, we need some mathematical model or need some computer coding skills in it. It's not very user-friendly for the industry. So currently we're working on to develop this computer user interface for a even simpler sample to answer analysis of the fish species using Raman spectra. So with this interface, you will be simply clicking the button here, load a Raman spectra, and all of the mathematical model I just introduced will do the calculation behind, either in the computer or in the cloud server. And after the calculation, the interface will be able to show you the results regarding which specific fish species your sample belongs to. So with this computer user interface, we can easily make it functional for the Internet of Things infrastructure to allow the instant information sharing along the supply chain with all the stakeholders. And for the second case, we try to miniaturize the method by using this microfluidic lab-on-a-chip platform. So it's a fancy term, but basically a microfluidic platform is manipulating a small volume of liquid in the micro-sized channels and chambers. And the simplest example of microfluidic platform you can imagine is this, I'm sure everyone is very familiar with it now. It's a lateral flow immunoassay that's used for the rapid testing of COVID virus. So I'm not sure if you try to disassemble this device, but there is a little strip inside. So the strip stores the reagents on it and also allow the liquid to go through. So you only need to apply a few microliter of the sample, and the liquid will go through the chip, go through the strip. So it's also manipulating the sample, the liquid in the microliter range. But this device doesn't have that complicated chip pattern design. So I say this is a simplest version of the microfluidic platform. And so with a proper design, microfluidic platform can integrate and tandem multiple assays onto one device. And that's also why this platform is also regarded as a lab on a chip. So when you're performing a complicated experiment, you might need to do several steps in the lab by using several different instruments. But with this lab on the chip platform, it's possible, if you design it properly, it's possible for you to complete everything on one device. And also these features make this platform to allow rapid and high throughput analysis. These devices are usually made with polymers or paper materials, which are quite cost efficient. Especially for the paper material, it's also very environmental friendly. So in this case, I try to develop a microfluidic lab on a chip platform for pomegranate juice authentication. So traditionally for pomegranate juice authentication, well, in this case, we decided to use DNA-based analysis because we think for the pomegranate, it has high chance to be adulterated with cheaper fruit juice such as apple and grape. And they have different genetic code. So we can work on the DNA difference. In the lab scale, several steps you will do. The first step is to extract the DNA from the juice. And then we'll need to do a DNA amplification. So in this case, instead of using a PCR, which requires thermal cycling thermal cycling to initiate the reaction, isothermal amplification just need a constant temperature to allow the reaction to happen. And then we also need a result visualization step. So the next step for my experiment is to integrate all these three steps onto one single device. So this is an illustration of how the device look like. So this is a 7-layer device. So the first 4 layers are made with paper. The black areas are wax. So for the filter paper we use is a super hydrophilic material. You will not be able to control the liquid flow with just the filter paper itself. But if you block the certain area with the hydrophobic wax, you will be able to constrain the liquid to only weaken or stay in certain area of the paper. And so this white areas are the hydrophilic area that we will allow the liquid to go through. And the bottom 3 layers are made of plastics. On the 4th layer here, we have a DNA extraction zone. So we will be loading our fruit juice samples onto this zone and use several layers of absorption pad at beneath to allow the liquid to go through but the DNA will be trapped on this device, on this area here. And then we will fold the first 3 layers on top and the last 3 layers beneath. So the stacking view look like this. DNA is trapped here. And after the DNA extraction, we will load a elution buffer from layer 1. It will go through all this layer and release the DNA from here into the 3 darker chambers at the last in the 7th layer. This is a layer for the DNA amplification reaction to happen. And in this analysis, we also designed 3 negative control area to serve as a quality control of this device. Since we're analyzing 3 different fruit species, so I developed this device to enable the multiplex analysis of all the 3 species of fruits altogether. And here's the representative results. So the brighter area, or in this case, we used a fluorescent dye to visualize the results. So the brighter areas are the samples, indicate that the sample contains the DNA, contains a specific biological species you're analyzing. While the darker area are the negative control to control the quality, to make sure the reaction is functioning properly. So compared to the conventional PCR-based method, this method using this microfluidic device with an isothermal amplification, we only need 1 hour versus the conventional PCR requires about 3 hours to finish the sample DNA extraction to the result visualization. And this device allows us to simultaneously analyze 3 types of fruit juice altogether at the same time. I also roughly estimated the cost. So for a lab scale manufacture of this device, it costs us about $4 per test. But I'm sure like for massive production, the cost can be further reduced. Oh, and one thing I forgot to mention is that if for this isothermal DNA amplification, we don't need a sophisticated instrument. So I simply assembled this a portable heating pad powered by a portable power, and then a temperature controller. So for this part of the heating pad, the assemble, I think, cost me about $20 and is completely reusable. So the overall cost of this analysis is way cheaper compared to more cost effective compared to the traditional PCR-based method. The result here is captured using a smartphone, using a camera on a smartphone. So it can also be easily integrated into the IoT system that we want to use for our smart traceability. So some take-home message for my talk today, food fraud is all about intentional misrepresentation of product for economic gain. And there are certain types of fruit that are more vulnerable to be adulterated. And I hope you will pay more attention to those food products with the specific features, like pay more attention to their suppliers to make sure if they're reliable. And then traceability system is the best prevention strategies for food fraud, but the food authentication analysis is really the last barrier to prevent food fraud, fraudulent products to enter the market. And if you combine these 2 strategies together, it can do an even better job with the help of the informatic tools. So with the smart traceability system, hopefully we can better safeguard the supply chain from the farm to the fork. Oh, and lastly, I'd like to share this website with you so you know where to report a food fraud when you suspect you have encountered one. So this is a website, and I think if you just search this title here, you will be directed to this website. And there is a little questionnaire asking where are you residing in. And so if you're residing in provinces out of Quebec, then CFIA will be the place you will report in your complaint the suspected cases to. In Quebec, I believe it's Ministry of Agriculture. That department in Quebec is dealing with the food fraud complaints. And with that, I'd like to acknowledge my supervisor, PhD supervisor, Dr. Xiaonan Lu, and my postdoc supervisor, Dr. Tao Huan, Dr. Terry Kerner, and my collaborators who also helped a lot with my experimental design. And definitely the funding agencies who funded my study and my research, the UBC 4-year fellowship, MyTax, Carleton Startup Fund, and also NSERC. With that, I'd like to thank you for your attention, and now opening the floor to any questions. Thank you.
Wonderful. Thank you so much. That was a great presentation. And just a reminder to all the participants, attendees, that you can type your questions in the Q&A button. If you go to the bottom of the screen in the Zoom call, there's a section you can press on Q&A. It'll pop up a window and you can type your question in there. So we already have a few coming in, so I'm just going to jump right into them if that's okay. The first one coming from an anonymous attendee. The question is, does the requirement for authentication of the fish fall to the distributor?
Does requirement of fish fall to? I think so. Wait, authentication. Sorry, you mean like, does a distributor need to provide any information?
Yeah, that's how I would interpret that. Yeah. Who's doing the testing?
Right. So I think that's a good question. That's the part that I'm not that familiar with, to be honest. But I assume like everyone in the supply chain dealing with the products need to make sure they're purchasing the product from a reliable source. Even though you don't spend money for doing the test, you should have some other strategies to make sure the product is authentic. It's authentic.
Right.
Yeah.
Yeah, great. Thank you. And also just side note, if anybody has a follow-up question for this, there's no limit on the number of questions you can post, so post away. Move on to the second question from Making Low. Hi, Dr. Hu. Do you make the extraction pad by yourself or was it bought directly?
The extraction pad, you mean the part that I'm doing the DNA extraction on the paper device, on the microfluidic device? So that is a commercially available material. The specific one I use is called Fusion 5. That is a material contains some glass fibers in it. So it's basically I'm using the silica material to retain the DNA. So I'm not sure if you're familiar with the DNA extraction cartridge. So usually they're using some silica-based material in there to trap the DNA. I'm using the similar strategy. Just instead of using the tube, I just use the material directly and assemble it together with my microfluidic device.
You built it all from scratch yourself?
Yes, the device was made from scratch. Yeah, but the material, like each part of the material was commercially available. We just need to design the pattern and then assemble everything.
Cool, thanks. All right, had another question come in from Jankun Wong. I'm a sushi lover, so am I. How much percentage of salmon are real salmon in those restaurants? That's
a good question. So I'm pretty new to Ontario. I did a market survey study when I was in UBC and surveyed I don't remember the number of sushi venues we visited, but we definitely collected quite a lot of samples from the sushi venues. And in general, we found like 23 or 22% of fish being mislabeled. The salmon was not a big hit for that. I think because in the Pacific Ocean, we have a lot of salmon. And I think people living in the coast area, they're very familiar with the appearance of different species of salmon. The pink salmon, it definitely looked different from sockeye. So salmon was not the biggest hit for the fraud in that area, but instead it's more of the snapper. So all of the snapper we tested was actually tilapia. So that was actually the biggest issue for the sushi venue, as far as I know, for the BC. And I read some news regarding the testing in Ontario. I think they had a lot of concern regarding tuna instead of sushi. So especially for the white tuna, there is no white tuna species specifically. It's just a description of the color, but there's no white tuna. And then there were testing showing some of the white tuna was actually escalar, which is an oil fish, can make you have severe diarrhea. Right.
Interesting. All right, we have another question also from Making Low. Usually food fraud analysis requires the specific contaminants to find ways to test. Basically, you have to know what you're looking for. If we don't know the contaminant in advance, how do you start the food fraud analysis if you need to do it, if required?
Right, that's a good question. So that will fall into the differentiation between the targeted analysis and untargeted analysis. So if you know what you're looking at, if you're familiar with a specific food product and you know it's commonly being adulterated with certain material, you will do a targeted analysis. But in the other case, especially when we're trying to compete the speed with the frosters, so they're also trying to develop new fraudulent method to circumvent our established method. So if in the case we want to compete with them, we will need to develop some untargeted analysis. So in that case, so the Raman spectrometer method is one of the example, good technique to use for untargeted analysis. Other good technique to use are like mass spectrometry. All of the methods that don't require extensive sample preparation can be a good source for the untargeted analysis. So in that case, you will collect the information of genuine products and you make a library of how the genuine product will look like. So whenever there's something new presenting in the sample, it will be picked up by the untargeted method. Even though you don't know what is specifically being included in there, you know there's something wrong. And then from there, you can do some further analysis to target that, to specifically analyze what is being included.
Great. Thank you. Any other questions coming in? I might ask one quick one. It's related sort of to the first one again. In my research area, I'm more familiar with the pharmaceutical industry where things like the FDA and those large government bodies have to approve every step of manufacturing. But I'm really a lot less familiar with food industry. So if a company's making a certain product and then they think, hey, we can make it cheaper if we add this or we take this out, is there any onus on the company to have to report that? Or can they make sort of just these unilateral changes to products, keep it on the market, and there's no, like they don't have to declare that change?
That's a good question. I think that's why we have the requirement for what to include in the label.
Right.
So you will need to label all the ingredients you use at a certain order from basically from the highest concentration to the lowest concentration, as long as that ingredient present at a certain threshold. Above a certain threshold, you will have to report that, include that in the packaging. And that's why like a lot of food fraud, we think it's more of or food like mislabeling, or we just call it a mislabeling because it's basically not labeling correctly the information on the package. It could be an unintentional practice because the manufacturer are not very familiar with the regulation, but that still causes a lot of downstream issues to the consumer.
Sure. Right. And those labels, the manufacturer has to pay for that analysis to make that label, right?
For some of the label, yes, like especially for the macronutrients, like what's the fat concentration, the sugar, the saccharides. But I also know that a lot of industries, they have a calculator. Just depending on what are the ingredients you use to manufacture your product, you can use a calculator to generate a rough information, and you can use that one as well.
Interesting. Huh. And sorry, one last follow-up for me. Is that calculator and that analysis, I imagine if you're talking about the ingredients going in, sometimes when you cook something, when you bake something, chemistry changes. So it's not necessarily representative of the final product if it's like a baked good. Is there any onus ever to look at the final product versus just ingredients going in?
Yeah, that's a very good question. Yeah, actually, a few years ago, there was a study. When I was in UBC, there's another group doing a testing on the sugar content in the bread. So most of the time, the label is based on the ingredients. But then during the baking, the sugar get caramelized or there's some changing. So you're actually getting less sugar in the final products. And I think that variation can be more than 10% or something.
Sure.
So it can be a huge one, but I think I'm not aware of a lot of industry doing that analysis for the last step.
Right.
Yeah, for the final products.
Yeah, and you can think of analogous. Sorry.
Right, food industry is not like pharmaceutical industry. Like if there's some deviation in the concentration there, it's not a super big hurt to the consumers.
Yeah, well, that's great things to think about. There's a lot of change that goes on in that. A similar comment coming in, I see now from Making as well. That's how the big companies do the cost saving. There's a lot of things that can't be covered by the ingredient label. That's absolutely true.